So caving lightly as a concept is the idea of not damaging the caves that we're exploring. Many of the features in the cave are super delicate, super fragile. They take tens of thousands of years or longer to form. So the idea behind caving lightly is you can go in, you can explore, you can see these beautiful sites, but you don't touch them, you don't break them. Whenever possible, you can see where a lot of cavers have gone before, so you stay in those confines so that we're not spreading our influence further out into the cave. So the idea is we can come in, we can explore, we leave a few footprints at most, we take our pictures, we take as much non-contact data as possible, and we leave the cave the way we found it. Any bodily excretions that you make in the cave, you have to haul those back out with you. Down here, there's not going to be the bacteria that's going to cause natural decomposition. So we really want to make sure everything comes out. You eat a candy bar, you're going to take that wrapper back out. If you're really on top of things and you find something a previous caver dropped by accident, you're going to pick that up and take it with you. Anything that is not native to the cave, that's not put there for a scientific purpose, goes back out. And so you try to pick up as much other stuff on your way back out to be responsible and help, because sometimes things just fall out of pockets and you might find a button that tore off someone's pants, so you'll pick that up and take it with you. Caves are unique. They are pristine environments that have lasted for tens of thousands of years. Especially some caves we think are going to be the best models for extraterrestrial habitation. How do you best describe a Mars lava tube? You find a similar environment here on Earth. So on the science aspect, it's important to preserve them the way they are so that we can better understand them as analogs for off-Earth missions. But just from the preservation standpoint, anything that we damage within the cave could be permanently damaged forever. We're talking about features that might take tens of thousands of years to form if they ever formed again. Some of our cave features may have stopped growing 1,000 years ago, and we don't know if they would ever repair themselves. So anything you damage, you've then damaged a piece of heritage that can never be remade again. So caving lightly for research's sake means that you leave the cave in a state of natural preservation. You're not introducing a large amount of contamination from human influence. Humans are innately dirty. No matter how much we clean, unless we actually come down here in a white bunny suit from a clean room, we're going to be shedding skin cells, we're going to be shedding bacteria, we're going to be shedding mold and fungus spores. That's going to happen. The hope is by caving lightly, we can minimize that impact. And then we set up contamination study sites throughout the course of the cave so that we can monitor exactly what is coming off of us as we're going to these deeper and deeper areas and seeing how pristine we're keeping things. Many of the native microbiology to topside terrestrial life are going to perish very quickly once they come down into the cave. The biggest risk is bringing something down here that's highly adaptable that could get established. And so that's why we plant the contamination studies, is to see, okay, did we bring something down that if it had a little bit of food, is it going to thrive or not?
It's kind of interesting because you can see that these two little red spots are definitely some bacterial. And then most of the ones that look really fuzzy, those are going to be fungal. So yeah, kind of interesting.
So for our spectroscopy work, we want to understand a lot of the super delicate fine crystals. The problem with the delicate fine crystals historically is those are the ones that are the most precious not to break. But they're also the ones that are the hardest to study without breaking and taking back to the lab to study. With some of the newer technologies, you can get a portable X-ray mineral device that is about that big. And you can bring that right up, very lightly touch the delicate crystal, take its spectrum, and you can see all the different metals that are in there without damaging the crystal at all. With our fiber optic spectrometers that we have, the whole thing is about the size of a lunchbox. It fits in our cave bag, and then we can use very fine fiber optics And that's the part that we lead right up to the samples. And so when that fiber optic is up there, is this tiny little viewport at the very end of our fingers with a light source right there held in the other hand. And that lets us make absolutely no contact because it's one type of light going in, typically ultraviolet, which is then causing the crystal to glow a visible color, green or blue or red. And we can measure the color in and the color out, and that's all we need to know in order to determine what is locked within the calcite crystals. So once we're in and out of the cave, one of the biggest things right now is worrying about contamination between caves. So one of the things we always do when we exit a cave is we clean the gear. When we're surfacing for the last time, we make sure we walk across a pad soaked in bleach to get any spores off our feet that we may have been walking around in the cave. We don't want to contaminate a new cave in that way. We make sure all of our knee pads and elbow pads go through a wash and sterilization cycle so that if we go into another cave, we're not going to bring samples from one cave into another cave and then cross-contaminate and possibly introduce microbes that were thriving in one cave that could just destroy the unique organisms in a different cave. You're not just leaving the cave in the state that you found it in, you're trying to make it better as you leave and leave it in a